Before starting up, please check if there is enough sheath fluid and cleaning solution, and whether the waste container is full. After finishing the check, turn on the instrument and computer power sequentially. If the waste container is too full, it needs to be emptied. If there is insufficient sheath fluid, please replenish it in a timely manner. After starting up, click on the maintenance interface to replace the sheath fluid and make sure that there are no air bubbles in the filter, syringe pump, and flow cell. Open the operating software interface, and the top right corner shows that it is booting up. Please wait for approximately 10 minutes until the boot up process is complete and the standby mode is displayed normally. Then, in a maintenance interface, click on the filter, plunger pump, and the rectifier chamber in sequence to remove any bubbles from each component. It is important to ensure that there are no bubbles in the previous component before removing bubbles from the next component. First, filter. Click on the maintenance interface and select the fill filter to tilt the filter so that the upper side adjacent to the pipeline is at the highest point. After hear 3 sounds, invert the filter and repeat the operation to remove air bubbles. Second, plunger pump. Click on the maintenance interface and select Fill Plunger Pump. When the plunger pump is sucking downward, shake it and push it upward to expel the bubbles. Gently tap the pipeline to remove bubbles. Third, rectifier chamber. Click on the maintenance interface and select Fill Rectifier Chamber to bounce the pipeline connected to the flow chamber and remove bubbles. Function Zone Introduction. First, measurement interface. Sample management area. Can choose 96 well or 40 well sample plate. Create new samples tubes in any position. Multiple tubes can be created under each sample. Can search and query sample plus tube files at any time. Import, export tubes or FCS files. Measurement control area can control experiment start and stop. Refresh experiment data. Control the entry and exit of the sample tree. Display onboard experiment data. Settings area can set tube measurement volume, control sample flow rate, modify experiment stop conditions and particle display count. Modify channel names, adjust voltage, threshold, and composition conditions. Data statistics area display particle count, percentage, absolute count, channel names, mean value, CV, and other data. Image management area can select various types of graphics, such as scatter plots, histograms, density plots, etc. Create different types of gates, such as cross gates, rectangular gates, elliptical gates, etc. Can zoom in, zoom out, and move images. Analysis interface. Compared to the measurement interface, there is a lack of a measurement control area in this interface, but all other functions remain consistent. Data analysis can also be performed in this interface. Quality control interface. It allows quality control experiments and displayed calibration operations, as well as the ability to query the quality control history. Maintenance interface. Enable instrument maintenance and self-diagnosis functions, allows for querying component lifespan and monitoring information of different functional parts. Setting interface. It allows users to set device instrument parameters, templates, and fluorescence channel switchings. Users can also modify particle display effects, perform user management, and adjust printing settings. After the completion of instrument startup maintenance process, you can proceed with the operation. On the measurement interface, click on the 40-hole sample tree. Select the corresponding number of wells according to the number of samples. Then create a new sample. Click on the In and Out button and place the corresponding sample tubes in the appropriate well positions. Once placed, select the template for the corresponding experimental project and apply it to the samples. Select the samples that need to be run and click on the Record button. After the sample is loaded onto the machine, observe if the first sample image is normal. Only proceed with subsequent sample experiments if it is normal. If it is not normal, stop the machine and check again for bubbles in the filter, plunger pump, rectifying chamber, and other positions. After performing maintenance and refilling, restart the machine. End the computer session. Export data in FCS file format or share the data in exported spreadsheet. Maintaining shutdown. Clean before shutdown. Place cleaning solution in position E7 and pure water in position E8. Inject the samples 5 times separately, high speed, 100 UL each. After cleaning, click the shutdown button in the upper right corner and wait for about 10 minutes. The display will prompt to turn off by pressing the power button. Turn off the instrument and computer power.